Thank you, Eric. We're going to start with a little film. Thanks for that, and all the people that participated. Welcome to our annual New York City Gala, celebrating Stuttering Awareness Week. Tonight we welcome the Chief of the Laboratory of Communication Disorders of the National Institute on Deafness and Other Communication Disorders at the NIH. He received his bachelor's in biology at the University of Wisconsin, and then his PhD from Harvard in genetics. What else? In 1981. His postdoc research, which was fascinating, was at the Howard Hughes Medical Institute, where he constructed the first full-length genetic map of the human X chromosome. That itself is extraordinary. Fortunately for us, he moved to the NIH in 1996, where his interest in stuttering was launched. His research interests are focused on applying the tools of human genetics and genomics to a range of topics that span the mission of the Institute. But his passion is stuttering research, and in 2010 he found the first 3 genes for stuttering, because that opened the door to everything. His current studies are focused on the genetics and neuroscience of stuttering. As a private citizen, he has served on the Stuttering Foundation board since 2003 for 13 years, and we have profited greatly from his vast knowledge. But tonight he's here in his professional capacity, representing the NIH and NIDCD, to give us an overview of his stuttering genetics research, including the genes they've recently found, the discovery of what the products of those genes do, and how this information has led to an understanding of the underlying causes of stuttering. Please join me in giving a warm welcome to Dr. Dennis Drainer.
Well, thank you, Jane, for the lovely opportunity to be here, and I'd like to thank everyone here for coming to help celebrate this great event. I've divided my remarks this evening into kind of 2 parts. The first part is a brief, hopefully punchy, and is the take-home message. Once we've covered the take-home message, then I'm going to sort of move on to some more recent things that are kind of interesting, we hope. So let me get right down to it. So this discussion is about basic research, where basic research in stuttering now sits. And of course, any discussion of research in stuttering, I think it's helpful to go over some of the more popular theories about the causes of stuttering. So bad parenting, always popular. There have been many versions of this over the years. Anxiety. So there's no doubt that anxiety plays some role in how stuttering presents. There's no doubt about it. And I think this comes from, it has ancient biological origins, but it's not something we understand very well, and it's not something us geneticists really have an entree to at this point. Nevertheless, another popular theory was what's called auditory feedback, hearing our own speech, right? We all hear our own speech, both by air conduction through the air and by bone conduction. And if there are problems with the way we perceive our own speech, it can cause problems with fluency. A very popular theory. Another one is timing or coordination between the two halves of the brain, okay? So it's been known for only about 150 years that a lot of speech functions are localized here in the left hemisphere of the outer part of our brain. It's called Broca's region, after Paul Broca, a famous French neurologist who had a patient who sustained a stroke and was fine, except he just lost the ability to speak. And Paul Broca actually convinced this individual to basically donate his brain to science, and on autopsy found a very specific localized focal stroke over here. Now, a lot of speech functions are localized here in the left hemisphere of the brain, but not all of it. So there are plenty of other things that go on in the right hemisphere of the brain, and one could imagine that these two halves, which if you just look at the brain, are mostly not connected to each other, if they have trouble communicating with each other, it could lead to problems with fluency. Okay, very popular theory. Basal ganglia. So this was always one of my favorites. The basal ganglia is a deep part of the brain. It's the part that goes wrong in Parkinson's disease. And people who, if you know a little bit about Parkinson's disease, one of the hallmark neurologic features of Parkinson's disease is something called tardive dyskinesia. If the people with Parkinson's disease, they begin to walk and they can't quite get going. And so it's a problem initiating movement. So this might sound kind of familiar because a lot of stuttering disfluencies are at the initiation of a sentence or a word, okay? So basal ganglia was always a significant possibility. And finally, a speech, of course, is a motor function, meaning it has to do with movement of our muscles, right? So there's a couple two major parts of the brain that are involved in motor function. There's something called the motor cortex, it's right up here. It's in the outer part of the brain. It's sort of a region here. And another part in the cerebellum, which is a thing that looks very different, sort of tucked underneath in the back. And both of those have very strong control over any movement. So motor problems. Okay, so these are some of the main theories, and I'll come back to these. Okay, so I'm going to talk just briefly about applied research. So sometimes this might be called translational research in the modern era. Things you might hear about, I just want to touch on. So people are all here on the same page. Deep brain stimulation, very popular for Parkinson's disease. They basically drill a hole in your head and they insert an electrode all the way through your cortex, down about this far into the basal ganglia, and they stimulate it with electrical impulses, and it seems to help. Okay, it seems to help. Another one, transcranial magnetic stimulation. This is kind of a new one. Imagine an extremely high-powered, very focused MRI scan. Okay, a magnetic, and when you get an MRI scan, it's a series of magnetic pulses. If you turn that up a long way, that magnetic field way up, and they actually design specific coils, depending on what part of the brain you want to target, they can actually direct extremely high-strength magnetic fields. And for some neurologic conditions, there is sometimes some improvement. It's not known very well how exactly they work yet. But there's some hope for this. People have tried TMS and stuttering. It hasn't yet proven to be an enormous cure-all. But the studies have been small. And then finally, something a lot of people may have heard about in the media. It was 2 weeks ago. And to be honest, the media didn't exactly present it correctly. They said computer-generated speech based on brain activity. And that's sort of true. What that is you take a an array of electrodes. It's usually about this big. And you go through a hole in the head. These are people who are already having brain surgery for epilepsy. So the neurosurgeons are already in there. They put this electrode on the speech parts over here, and they just ask the person that these people are conscious, okay? So there's no pain receptors in the brain. And they ask them to read. Read and read and read and read. These electrodes just recorded all the activity. Then they gave all that to a computer and asked the computer to find patterns in the electrical activity, okay? And so it's kind of a computer interpreting the electrical activity of the brain to produce speech, kind of, okay? But these are people who are already perfectly fluent, is caveat number one. And caveat number two, how good was the computer? Got 43% of the words right, okay? So that's the latest on that. I think that is exciting, but of course everybody here can probably decide for themselves whether they think drilling a hole in your head to put something into your brain is going to be the way to go to address stuttering. Okay, so I want to also mention, people often say, well, what about just regular brain imaging studies of stuttering, like MRI scans or PET scans or something like that? There are lots of these, okay? There are 29 studies published to date, lots of different types of imaging, mostly MRI. Most show alterations over here on the left side, over here in the Broca's area. The problem is that you can't tell whether those differences are the cause of stuttering or the result of stuttering. That's the problem. And the other problem is these studies cannot see down to the level of cells and molecules. Okay? And as a one-time veteran of the pharmaceutical industry, I'll just tell you that pharmaceutical science operates at the level of molecules. Okay? So you've got to get down to that level if you're going to be finding targets that are specific for this particular condition. All right. So, what about genetic factors? Well, it turns out I won't bore you with the evidence that stuttering is genetic. I'll just say it comes from twin studies of identical and fraternal twins. It comes from adoption studies. It comes from other kinds of studies. But none of this data is ours. It's accumulated over 40 years, and the evidence, I would say, is overwhelming at this point, that there are some sort of genetic, inherited causes. So we have now identified 4 genes in which mutations cause stuttering, and that's most of what I'm going to talk about the rest of this evening. It only took 15 years. It turns out there was no reason to expect this, and in fact we were surprised to find that the products of all 4 of these genes participate in the same function inside cells, and they all interact with each other. They all interact with each other. So just by genetics alone, we kind of stumbled into a number of things that are all sort of a piece in terms of what they do inside the cell. All right, so what are they? Well, they're genes that code components of what's called intracellular trafficking. Intra, inside the cell. So this is a fundamental process that's in all cells of the body. All cells take things in, they move them around to where they need to go. Sometimes they send things back out. But cells have a lot of internal structures, and things need to be directed very carefully to the right place. All right? So I'm sorry for this very sciencey diagram. I got this off the internet. The people who discovered all this was the subject for the Nobel Prizes in 2013. The machinery, things are taken into cells, and they go through these compartments called endosomes. Endo meaning inside. Soma meaning body. These are bodies inside the cell. They're different compartments, is the way you could think of them. They're early endosomes, late endosomes, things called lysosomes. And the machinery that moves things around, it appears to be what is wrong in the people who have mutations in the first 4 genes that we've found. Okay, so it's probably no surprise that anything in biology can go wrong. And in fact, my attention was, I just picked this one out because this was the actual paper that first caught my eye. People began to realize that disorders in this trafficking business, in fact, are common in neurologic diseases. At first, trafficking disorders were found for very, very rare disorders, diseases I'd never heard of, things like Hermansky-Pudlak syndrome, or Perry syndrome, or Niemann-Pick type C, rare medical genetic disorders. But more recently, trafficking deficits have been found in much more common disorders, and I'll get to that in a minute. So we wanted to try and get something going in this business. So Bettina Winkler, who's a cell biologist from the University of Virginia, and I organized this meeting called Neurologic Disorders of Intracellular Trafficking. The attendees, 25% geneticists, like me, 25% cell biologists, people who actually understand trafficking, 25% clinical neurologists, and 25% drug developers, people from the pharmaceutical industry who attended this meeting. We had people from 13 countries, and it was a great meeting, but it was really cold. It was in January in Colorado. Okay, so while these people weren't attending the meeting, it turns out that the idea of neurologic disorders of trafficking, intracellular trafficking, have not gone unnoticed by the drug developers. So this guy, Joachim Hertz, who's at the University of Texas Southwestern, a big, high-powered medical research institution, published this paper recently. It's the most jargon nut, I promise. It's never going to get any worse than this. Reversal of ApoE4-induced recycling block as a novel prevention approach for Alzheimer's. So from the abstract, ApoE4 is the most present. So ApoE is a blood protein, and it comes in 4 different flavors, and different people have different types. Okay? If you have E4, there's E1, E2, E3, and E4. If you have E4, you are going to have to worry about getting Alzheimer's disease. It's the largest single risk factor for Alzheimer's disease. Okay? It's the most clinically important risk factor for late onset Alzheimer's disease. Available evidence suggests that the root cause of this increased risk is a trafficking defect at the level of the early endosome. So this was like, wait a second, where have I heard this before? So what these people did was they actually ran a pharmaceutical screen. They actually started screening compounds to reverse this in nerve cells from patients with ApoE4 Alzheimer's. And they screened 10,000 compounds, and they found this one, which just has a code name. Now, people here who don't know Sander, Sander Flom, of course, has an incredibly distinguished career in the pharmaceutical industry, and he can tell you that a hit in a compound screen is a far cry from an approved drug. And in fact, of course, this is something for Alzheimer's disease, not stuttering, but it is directed at correcting a trafficking deficit. And if there are smart, good people who are interested in using this relatively new area to develop new therapeutic approaches to neurologic disease, then where does that get us? Well, so our take-home message here for today is genetic studies are identifying the causes of stuttering at the level of molecules and mechanism inside cells. At least some stuttering is caused by a mechanism that is shared with other neurologic disorders, a trafficking deficit, including some very common, very economically important neurologic disorders. So drug developers have recognized that this is a new entry point for development of novel drugs for neurologic diseases. And another principle of pharmaceutical development is oftentimes very, very successful drugs were a surprise. They were developed for something else and they were discovered to be useful in another indication at some point later, all right? So the goal here is if there are lots of people now interested in developing drugs for trafficking disorder, the goal would be to have a rising tide float all boats, including the stuttering boat. So that's today's take-home message. I think if people will just grant me a little patience, I'll tell you about some other kind of wild things that have been going on in the lab. We're interested in the animal model. We're interested in finding affected cells in the brain, and I'm going to tell you about that. And we're interested in finding affected brain structures for what I think is sort of a new look at brain imaging at this point. So let's talk about an animal model. An animal model is a problem because animals don't talk, right? But one thing that I believe about stuttering, when I started to learn about it, it's remarkable what's not wrong with the speech of people who stutter. People who stutter don't have problems with memory or what's sometimes called word finding. They don't have problems with grammar. They don't have problems with syntax. For the most part, they don't have an articulation disorder like a lisp or something like that. People who stutter know exactly what they want to say. They just can't say it at the rate they would like. Okay, so I think calling it just a speech disorder is a little, it's just not, it's not sufficiently precise. It's really a disorder of the voluntary control of speech, the rate of speech production. That speech can otherwise sometimes be quite normal. Everybody knows that I learned the joke from the stuttering community that no one stutters when they talk to their dog, and it turns out to be largely true. So oftentimes the speech of even relatively severe stutterers can be quite normal. So maybe the mouse could be a model for the voluntary control of vocalization, even though the mechanisms of their vocalization are very different. They display extremely rich vocal communication, mice do. Much of it is ultrasonic. It's way too high for us to hear. All the interesting stuff we can't hear. Somebody was interestingly pointed out that all the complicated vocalization in the house mouse in mice, it's the frequency, the higher the frequency, so the frequency starts at the highest frequency, just beyond the highest frequency that cats can hear. Just beyond. It starts at about 25 kilohertz, 25,000 hertz, which is, there might be some young person here who can hear that high. I haven't been able to hear it in years. Okay. So we engineered human stuttering mutations into mice, and I'll tell you what happened. Those mice grow and reproduce normally. They exhibit normal behavior in a very wide variety of settings and tasks. These mice run around. They do all the normal things. In fact, much of their vocalization is completely normal, except they have these gaps in their vocalization. If you take mouse vocalization and you lower the frequency so that we can hear it, it kind of sounds like a bird. It's like chirp, 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 chirp. Chirp, 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 chirp. That's sort of what it sounds like, okay? So it comes in bouts, and then there are silent spaces in between the bouts. The mice who carry human stuttering mutations have problems in all of it. It's like chirp, 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 chirp. So they have more gaps, and they have longer gaps. But everything else about their vocalization is actually fine, okay? So if you take the speech of the humans from whom those mutations came, and you give that MATLAB program, you give that acoustical analysis program, the speech of the humans instead of the mouse, they find the exact same thing in the speech of the humans. That's exactly the same problem. Okay, so what can you do with a stuttering mouse? Well, an animal-wide exam of the tissues by traditional methods showed nothing wrong with this mouse. Nothing. We gave it to the best guy at the NIH. He couldn't find a thing. And it's not surprising because there's hardly anything wrong with this mouse, right? But my postdoc, Taeyeon Han, said, no, we're going to look harder. So he started doing detailed cell staining studies to get different stains, to look at all the different kinds of cells in the brain, or at least as many as he could buy, all right? And most cell types in the brains of these mice were normal, but a significant and very specific problem was found within astrocytes, which is a type of support cell in the brain. It's not an electrically active neuron. It's what's called a glial cell. Glia is the German word for glue. It kind of holds the electrically active neurons together and sort of does care and feeding. And these things are most pronounced. Astrocytes are distributed in many places in the brain, but they're particularly concentrated in what's called the white matter. The white matter are the cables sort of the long distance cables between various parts of your brain, are connected by white matter. And that's where the astrocytes really like to hang out. So these problems were most pronounced in something called the corpus callosum. So what's the corpus callosum? It's the main structure connecting the two halves of the brain, and as I mentioned, it's very highly enriched in astrocytes, okay? So this is the human brain looking down from above, okay? And if you kind of pull it apart, this is a very old, famous drawing from 120 years ago. But this thing, this big cable, this white matter cable, that connects the two hemispheres of the brain is the corpus callosum. All right? So, what we understand now, while there are more genes to be found, the genes identified to date point to a disorder of intracellular trafficking, which is an emerging concept in neurologic disease. Astrocytes appear to be the primary cell type in which this pathology is manifest, okay, where this shows up in the brain. Astrocytes are particularly important in the connections between the hemispheres of the brain, particularly in the corpus callosum, and may represent the site of a major pathology in stuttering. Okay? So, I'll tell you, back to the main theories, I think, other than, of course, rejecting bad parenting is a likely possibility, I put this one at the bottom of my list. It just didn't seem as compelling as some of the other possibilities. But sure enough, what we found so far looks like that's what's going on. Okay? So we have a long way to go, but I'll just end things by saying that I have breezed through 20-some years of work that was actually done by a host of people, a very large number of incredibly talented people, to whom I owe an enormous debt of thanks, because it wouldn't have been possible. Of course, our stuttering research subjects are the foundation on which all of this is built, and we have lots of individuals and organizations, but of course the Stuttering Foundation of America is sort of, now it's just called the Stuttering Foundation. Right. Sorry. The Stuttering Foundation, I need to update my slide, has sort of always been there. On a personal note, I'll just say that, biomedical research is a slow and frustrating business. It just is. You're constantly going up blind alleys. You don't really know oftentimes where to go. I mean, you've got some possibilities, but they all look about as good as each other. So as we have encountered various struggles over the course of the last 20 years, the fact that we had the support of the Stuttering Foundation always made an enormous difference to me personally. And I think it carried me through knowledge of the support of the foundation and Jane. Carried me through many a difficult hour in this research. So I just want to end with that and say thanks to everyone, and I'll be happy to take any questions.
Fans, I have a question. Medication, based on your experience for people like us, the basic stutterers, is there any medication that you have come across before one makes a speech or gets up that one should take before?
So many things have been tried. Anxiolytics, so things that tend to reduce anxiety, almost always help some, at least for a while. The problem with a lot of anxiolytics is, of course, people can develop tolerance and dependency and all sorts of problems. So they are often not so successful in a long-term regular usage. But if there was some really important thing, I think anxiolytics could be very helpful. Now there are people, Dr. Gerald McGuire at UC Riverside is a psychiatrist and a person who stutters. And he's very interested in this question. He's tried a lot of things. And I think some of the people who he has seen have experienced some benefits. But the gold standard in a drug trial is a double-blind, prospective, placebo-controlled trial, and no medication for stuttering has ever passed that standard.
Beta blockers.
Beta blockers, yeah. Many things, so I should also point out, because I'm a cynic, many things work well right at the beginning for stuttering. Many things. And then their effectiveness fades. And so it's not clear whether, when something works at the beginning, whether it's actually specific to what that treatment is, or whether it's just this general phenomenon that all sorts of things sort of work at the beginning some. So that's my cynic's view. Yeah. Yeah.
Like alcohol?
Alcohol? Absolutely.
But I wanted to ask about the mice. Maybe you said this, but did you discover that some had the blocks, or did you induce this?
No, they have this all on their own. Yeah, so mice have all this rich communication. We used a very particular kind of vocalization that's extremely specific because we could control it in the lab, and it's instinctive. Okay? So they're called pup isolation calls. If you take a little mouse, like a few days old, and you take it out of the nest, okay? After about 90 seconds, this little mouse starts producing these things. And they're stereotyped, and they're extremely robust. They're actually loud. They're too high for us to hear, but they're amazingly loud for that little tiny mouse. And the mother finds it and retrieves the mouse and brings it back, okay? And those things are known to be under strong genetic control, and we can induce them anytime we want in the lab. They stop doing them as they get older. They only do them in the first 10 days of life. And so another caveat is this is not communication. The little mice don't develop hearing until day 10 or 12. So they can't even hear when they're putting these things out. It's been described as an infant just crying, you know, a newborn crying. But they are extremely reproducible, and if you put the human stuttering mutations in, of course you get a litter of mice. The way the laws of inheritance work, some of them get the mutations and half get it and half don't. It's just the way it works. We don't know which animals have the mutation and which don't, because they're too little to get a little bit of tissue from to look at their DNA. So we just record them all and ask the computer to tell us if there's a difference. And then we determine which ones have the mutation and which ones don't have the mutations later. But it's an instinctive, very stereotyped vocalization, and the animals who have the mutation have the problems, and their litter mates who don't have the mutation don't have the problems.
Do you think that the drug companies have a good economic incentive to work on this problem?
Repeat the question.
Yeah, so this is a very good question. It's a question, do drug companies have the economic incentive to do this? Right, it's a large problem worldwide, and I think there would be demand for this sort of thing. We all know that a new chemical entity from scratch is developing an approved drug is an enormously expensive undertaking. And so what's been done in the past is to try previously already approved drugs for this new indication. That's been the approach in the past.
Thank you so much. That was so fascinating. I have lots of questions, but I'm only going to say one. I was interested to know, you mentioned that the trafficking issue seems to be present in other neurological disorders that are more common also. I'm wondering what they are.
So it looks like probably the big player here is Alzheimer's disease, but a disease called Huntington's disease, which is a genetic disorder, is almost certainly a trafficking disorder. And there are some others. It looks like maybe some forms of some forms of Parkinson's disease. Because Parkinson's disease is probably not just one disease. There's slightly different flavors. Almost certainly, some forms of Parkinson's disease are trafficking disorder. Probably. Yeah.
Well, thank you, Dennis. You know, your work,
Sorry.
your work around the world gives us all hope. But before you go, we understand that the NIDCD will host a day-long symposium to celebrate your retirement at the end of the month. And so we would like to be the very first to wish you a wonderful, much-deserved retirement, and are excited to know what you're going to do after retirement, because we're counting on you then.
Well,
Okay?
priority number one is to stay on the board of the foundation.
That was a wonderful presentation. Thank you so much. Thank you all for being with us tonight.